Hello students, I welcome you to the EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, Karnal in Haryana, India. Dear students, during evolution of the enzymes, functionally significant residues are conserved more than the rest of the residues in the sequence. That is, in an enzyme, these residues which are involved in major function, they are conserved more, and the residues which are not conserved, they may not have some major role. The conserved residue may be present singly, or it may be present in contiguous short region, what we commonly call is a motif. The residues inside the active site of an enzyme are conserved for functional roles such as binding and catalysis of the substrate into the active site. The catalytic residues are the actual reactive amino acids and the stabilizers of the transition state. In addition, conserved glycines and proline residues outside the active site are conserved for the accurate positioning of the actual active site in the 3 dimensions of the enzyme structure. In module 6 to 10, we have seen that pairwise sequence alignment is used as a similarity search tool to find similar sequences in the database. This was to identify the members of a family so that they actually are then prepared as a data set for belonging to one particular family of the enzyme for performing one single function. Pairwise sequence alignment allows drawing structural, functional and evolutionary relationship between 2 sequences, whereas to extract the actual functionally significant residues from the sequence information, a conclusive inference as to the consensus residues among several sequences need to be drawn. Therefore, during evolution, mutations occur to enable species to survive under different changing environment, but the functionally significant residues are conserved along with their 3-dimensional positions so as to form the active site correctly for catalyzing the same chemical reaction in all the species by the same enzyme. Therefore, alignment of several enzyme sequences for the same reaction is useful for detection of the conserved residues to identify their functional roles. The alignment of several sequences is known as multiple sequence alignment, that is an MSA, which allows us to detect conserved residues which are otherwise hidden in the pairwise sequence alignment. In addition, detection of conserved residues outside the active site gives an insight that how they are involved in the folding pattern in the proteins. Detection of conserved residues within the active site and outside the active site may lay the foundation to develop an initial set of peptides for developing a XR model for structure-based drug design. Further, an MSA is a prerequisite for constructing the phylogenetic tree, reflecting the evolutionary divergence over time. Phylogenetic tree, therefore, may be used to select closely related multiple template structures for interactive homology modeling of the protein sequences. Consequently, an MSA has several important applications, and in this module, we'll focus on detecting the binding site residues involved in determining the specificity and detecting reactive catalytic residues involved in determining the turnover number of the enzyme, as well as detecting significant glycines and prolines which are required for accurate protein folding, and above all, an MSA is a prerequisite for constructing a phylogenetic tree. Therefore, the objectives in this module on detecting active site of enzymes begins with the compiling a set of enzyme sequences from various species known to be related to each other either closely or distantly for construction of multiple sequence alignment. The constructed multiple sequence alignment will be displayed graphically to extract maximum amount of information hidden in multiple sequence alignment for detection of functionally significant residues. Finally, we'll use multiple sequence alignment for construction of the phylogenetic tree. In this module, we'll take example of HPRKP, which is a bifunctional enzyme. HPRKP as kinase accepts HPR and pyrophosphate as substrates to produce serine phosphorylated HPR and inorganic phosphate. As phosphorylase, HPRKP accepts serine phosphorylated HPR and inorganic phosphate as substrate to produce unphosphorylated HPR and pyrophosphate. We'll detect the functional residues of HPRKP in the present example from multiple sequence alignment. For detection of functional residues of HPRKP, we'll use multiple sequence alignment. As 3D structure of HPRKP bound to its substrate pyrophosphate is not available, we'll compile a set of closely and distantly related HPRKP sequences for construction of multiple sequence alignment. We can use BLAST searches for compiling a complete set of closely and distantly related HPRKP sequences. However, in this example, we'll use keyword-based search of reviewed Swiss prot database at UniProt knowledge base. Although BLAST searches at close, intermediate and distantly diverged sequences using PAM30, PAM120 and PAM250 levels will lead to the same set of sequences, but here we'll use this new approach because we have available reviewed Swiss prot database, and in this database we have the entries which can be relied upon for construction of multiple sequence alignment. For compiling reviewed Swiss prot entries using keyword search, visit UniProt knowledge base and search for HPR kinase phosphorylase. This search returns 215 reviewed Swiss prot and 4,512 unreviewed Tramble sequences. Swiss prot is the manually annotated component of UniProt knowledge base. It contains manually reviewed and annotated proteins with information extracted from literature and also curated evaluation for sequence analysis. Tramble, on the other hand, is computationally translated protein from nucleic acid sequence database as EMBL, that is European Molecular Biology Laboratory, which are still awaiting review for inclusion in the reviewed Swiss prot database. Reviewed 215 entries is sufficiently large data set, therefore click reviewed 215 hyperlink to display reviewed entries. By default, UniProt knowledge base results display 25 entries per page. We have 215 entries, therefore to display all 215 entries in a single page in your web browser, select 250 from the drop-down list to show 215 entries in a single web page. At this stage, we need to analyze the lengths of the sequences to be included for constructing multiple sequence alignment. Therefore, sort these 215 sequences in increasing order of their length by clicking on the down arrow button in the column labeled length. In this sorted list, it is clear that first sequence of 216 is of very short length as compared to the next sequence with 301 residues. Therefore, this sequence will not be included for multiple sequence alignment. Next to 301 length sequence, the sequences continue incrementing in lengths of 1 or 2. Therefore, this represents a genuine variability in sequence length during natural evolution. Scrolling down The sorted list shows that sequence lengths differ by 1 or 2 residues. However, the last sequence again has unusual length of 615. Therefore, this sequence will not be included for multiple sequence alignment. On the top of the list, click on the button in the column labeled Organism to sort the sequences in alphabetical order by the organism name. This will help in locating a sequence in multiple sequence alignment by scrolling down the organism name. Now select all the sequences by clicking checkbox in the first column heading. Now find the sequence with length 216 and unselect it. Similarly, find the sequence with unusual length of 615 and unselect it. This compiles our set of sequences to be used in multiple sequence alignment. For constructing multiple sequence alignment from this page, click Align button in the toolbar at the top of the page. This will begin alignment using application tool Clustal Omega. Clustal Omega is a new general purpose multiple sequence alignment tool which can align up to 4,000 sequences or a maximum file size of 4 MB. The appearance of alignment result may take some time. For more help for Clustal Omega, kindly visit the help documentation. Also visit Clustal Omega frequently asked questions page. The results of the multiple sequence alignment will appear in the browser window. Each sequence is written in successive rows with aligned residues in successive columns. Each sequence is written in a separate line with the residues displayed in 60 columns. The residue in a column may be identical, representing conserved position in all sequences, or may be similar as per physicochemical properties, representing semi-conserved positions, or even it may be a gap which represents insertion or deletion during evolution of an enzyme or protein. When a gap is inserted, a hyphen symbol is displayed in its position. The results page of the Clustal Omega alignment allows highlighting alignment features based on annotations and amino acid properties. To highlight aligned residues on the basis of conservation and similarities, click the first checkbox under amino acid properties. This will highlight the conserved columns having identical residues with dark gray background. Columns having strongly similar amino acids with gray and weakly similar columns with light gray background. Otherwise also, after the last sequence in the alignment, consensus pattern is displayed in one of the 3 consensus symbols. A star symbol in a column indicates the presence of the same residue in all the sequences. A semicolon indicates semi-conservation of the residue with strongly similar properties. And on the other hand, a dot or a period indicates residue with weak similarity present at these positions. The conserved identities in the multiple sequence alignment are clear to draw an inference for their participation in the significant function of the enzyme. Similarities, either strong or weak, however, convey allowed mutations. A single replacement is sufficient to classify it as a non-identity. Therefore, similarities do not quantify the extent of replacement. That is, the consensus representation does not convey the frequency of replacement. But to draw meaningful conclusion for the involvement in a significant function, there is a need to convey the extent of replacement also. The graphical representation of the extent of conservation and replacements in a multiple sequence alignment can be shown as 100% stacked bar, or sometimes called 100% stacked column diagram, and it is commonly known as sequence logo. A sequence logo or 100% stacked column bar diagram compares the percentage that each amino acid contributes to the total at each position. Therefore, use 100% stacked column diagram to emphasize the proportion of each amino acid in a given position in protein sequence. The proportion of each amino acid is displayed using vertical rectangles with heights corresponding to the proportion of each amino acid. It can be drawn using any spreadsheet application such as Microsoft Excel. In this set of hypothetical sequences, the first sequence is shown to have each amino acid in alphabetical order along sequence length. The second sequence replacing the second last residue with a tyrosine, followed by the third sequence replacing third last residue with a tyrosine, and continuing mutating in this way till the first residue in the first sequence is replaced with a tyrosine. This is shown to be aligned hypothetically in the way shown here. The amino acid composition at each position is then entered in spreadsheet with columns labeled 1 to 20 for amino acid positions and rows labeled for amino acid symbols themselves in order. After entering amino acid composition at each position, select stacked bar column command from toolbar in the insert menu of MS Excel application. This will display 100% stacked column bar diagram, and this diagram clearly shows 95% alanine and 5% tyrosine in the first position with corresponding heights of the rectangles. Similarly, it shows 90% cysteine and 10% tyrosine in the second position. The complete vertical rectangle in the last column reveals the conservation of the tyrosine at this position. Therefore, a sequence logo or 100% stacked column bar graphical display reveals the extent of conservation and replacements in a multiple sequence alignment. For constructing sequence logo, let us download multiple sequence alignment of HPR-KP by clicking download button in the toolbar at the top of the page at UniProt KB Clustal Omega multiple sequence alignment. This will present a dialog box to select format and mode of downloaded file. This saving of the MSA in FASTA will help in further offline analysis for highlighting identities at different levels of conservation such as 99% or 95% or any other level using multiple sequence alignment editing applications such as BioEdit. The alignment results will appear in browser window in FASTA format, sometimes called multi-FASTA. Select all, copy on clipboard, paste in notepad and save with the chosen file name with .fasta as extension name and save as type all files selected. Visit web logo at 3plus1.com and upload the saved alignment to create sequence logo. A sequence logo describes the information content of the alignment. Here, sequence logo in this diagram for positions 1 to 180 is shown in 3 rows with 60 positions for each row. Underneath each row, consensus sequence of Clustal Omega is pasted manually by me. The x-axis shows the position of the alignment. The y-axis describes the amount of information in bits at a particular position. At each position, there is a stack of amino acid symbols occupying the position. The overall height of the stack represents conservation of amino acid at that position. Higher or big stacks, that is, stacks with more overall height represents higher conservation at that position, and positions with small stacks represent higher level of mutations of the amino acids at that position. Large symbols within a stack represent frequently observed amino acid at that position as compared to others in the same position. In addition, there is a wave of information from beginning of the sequence position to the end representing the change in information along sequence. This means that all stacks do not reach the same height as in 100% stacked column bar diagram. The calculation of stack height at a given position is influenced by the amount of conservation present at that position. This shows that information at each position is not same. Higher stacks falls within positions having conserved and strongly similar residues. Here the sequence logo for the positions from 181 to 240 is presented. We find a stretch of higher stacks in the first row, and this actually corresponds to conserved motifs and a large stretch of similar residues in HPRKP. This is actually the active site of the enzyme. Therefore, higher stacks in a given stretch indicates the presence of a binding or catalytic site. Another sequence logo application available at Center for Biological Sequence Analysis at Technical University of Denmark DTU allows construction of many types of sequence logos. Try for construction of Shannon type sequence logo using previous hypothetical sequences set to understand the interpretation of this Shannon type sequence logo. The Shannon type logo is somewhat similar to the one constructed at weblogo at 3plus1.com. The x-axis again shows the position in the alignment. The y-axis also describes the amount of information in bits. At each position, again, there is a stack of amino acid symbols occupying the same position. But instead of displaying the residues actually observed in the alignment, Shannon type logo includes other residues predicted with information theory. Here, this may be considered as mutations which may be expected in future during a further evolution. However, biochemists may test these suggested mutations in the laboratory through site-directed mutagenesis. The suggested mutations in the conserved motif containing active site of enzyme HPRKP may be tested in the laboratory through site-directed mutagenesis followed by expression of the mutated HPRKP and purification of this mutated HPRKP for activity measurements. For example, mutation of conserved dipeptide aspartate to suggested glutamate may be tried for testing activity of the enzyme. Now let us analyze multiple sequence alignment using cluster omega consensus notation. A semicolon in the last column of alignment in this example shows that it is conserved with strongly similar properties. This indicates the replacement of residue with other residue having similar properties. For example, mutation between methionine and leucine residues in the present case. Cluster omega defined 9 groups of strong similarities and 11 groups of weak similarities. For example, strong similarity is indicated for the group methionine, isoleucine, leucine, valine. When any of these residues replaces each other in a column, then it is considered as strong similarity. However, in case phenylalanine may also exist at a position where any of methionine, isoleucine, leucine, valine may be present, then this strong similarity is considered as a weak similarity. So therefore, there are groups for the strong similarities and for the weak similarities. From now onward, the sequences from HPRK underscore PADE from Pediococcus pantosaceus to HPRK underscore stack C from Staphylococcus aureus will be displayed. The aligned 60 residues reveals a mutation having strongly similar amino acid such as phenylalanine, tyrosine and tryptophan, the aromatic group of residues. Such strongly similar mutations indicate participation in packing residues inside the hydrophobic core of the enzymes or the proteins. In addition, 3 columns show dots in the consensus. This indicates presence of weakly similar residues as in the second column showing glycine and asparagine. In addition, Two more columns show dots in the consensus. This indicates that these weakly similar positions are allowed to mutate with less similar residues for optimizing enzymatic activity in different species having or surviving different environments. Therefore, these weak similarities may be significant for developing an initial set of proteins to be used for developing a CSAR model, that is quantification of structure activity relationship model, for structure based protein or peptide design for engineering enzyme activity under a given environment. Next 60 aligned residues reveal the presence of conserved histidine in all strain analyzed. This is a strong proof for the involvement of histidine in reaction mechanism or forming a salt bridge burying a negatively charged residue such as aspartate or glutamate inside hydrophobic core of the protein, or forming an ion pair on the surface for correct three-dimensional folding of the protein. The experimental knowledge confirmed the participation of this histidine in the reaction mechanism. Therefore, Clustal omega was able to predict or detect this histidine correctly. Next 60 residues reveal presence of short conserved sequence of length 4, that is a conserved motif comprising of glycine, lysine, serine and glutamate. This conserved motif is a strong indicator of active site or the binding site of the HPRKB. The experimental knowledge confirmed participation of this conserved motif in the binding of the substrate for the catalysis. Further, conservation of two consecutive aspartates indicate participation in forming the active site by these residues, and the experimental knowledge confirmed participation of these aspartates in the reaction mechanism. Conservation of 3 single residues, 2 glycines and 1 arginine is also displayed. Conservation of charged residues such as arginine is a strong indicator for involvement of arginine in the reaction mechanism or for burying some negatively charged residues such as aspartate or glutamate inside hydrophobic core of the protein by forming a salt bridge, or exposing an ion pair on the surface for correct 3D folding of the secondary structures. The experimental knowledge confirmed participation of this arginine in reaction mechanism for stabilization of the transition state oxyanion. The conservation of glycine residue is a strong indicator of glycine being part of a turn in three-dimensional structure of the protein. We look at single conservations and similarities of the uncharged residues in the next module. Next aligned 60 residues reveal conservation of 2 glycine residues, indicating their participation in a turn in three-dimensional structure of the protein. But we look at single conservations and similarities of uncharged residues in the next module. Next aligned 60 residues reveal conservation of a glutamate, indicating its involvement in reaction mechanism or forming a salt bridge or forming an ion pair on the surface for correct 3D folding of the enzyme. However, the enzyme experimental knowledge of the enzyme confirmed participation of this glutamate in burying a positively charged histidine residue inside the hydrophobic core of the protein. Similarities in hydrophobicity, polarity and isoelectric point can be represented through periodic spectrum of amino acids. Alanine is minimal hydrophobic with increasing hydrophobicity both up the periods and left to the columns up to the phenylalanine. The hydrophobicity of polar side chains increases from glutamate both up and right up to tyrosine. This spectrum provides residues for packing the hydrophobic core of the proteins. The isoelectric point increases periodically from acidic residues to arginine through increments of approximately 2 units. This provides residues for acid-base catalysis from pH 3 to 13. This periodic spectrum provides for mutating similar physicochemical amino acids. For example, clusteral omega groups methionine, isoleucine, leucine, valine as distinct group, allowing them to replace each other, having strong similar physicochemical properties. Similarly, F-phenylalanine, tyrosine and tryptophan is another group with strong similar physicochemical properties. The consensus notation of HPR-KP multiple sequence alignment reveals 2 conserved motifs, 7 identities, 22 strong similarities and 11 weak similarities. In total, there are 33 similarities which are due to mutations by residues having similar physicochemical properties such as similar hydrophobicity, similar polarity or similar isoelectric point. We have seen that HPR-KP in vitro as kinase accepts HPR and pyrophosphate as substrates to produce serine phosphorylated HPR and inorganic phosphate products. As phosphorylase, HPR-KP accepts serine phosphorylated HPR and inorganic phosphate as substrate to produce dephosphorylated HPR and pyrophosphate as products. From experimental observation, it is known that residues from glycine 151 to glutamate 159 forms P-loop for binding ATP or pyrophosphate as substrates. The residue histidine 136 and aspartate 174, aspartate 175 are catalytically reactive, and residue arginine 202 is involved in the stabilization of the oxyanion transition state. The conserved motif in P-loop and reactive diaspartate as well as reactive histidine 136 and transition state stabilizer arginine 202 are predicted correctly from multiple sequence alignment in the consensus sequence by clusteral omega. In 3 dimensions, serine 46 of HPR and arginine 202 of HPR-KP are present on the opposite sides of the phosphate group. Histidine 136 and 2 aspartates are present near the serine 46 of HPR also. Therefore, identification of conserved residues can be used for detecting functionally important residues in enzymes using this clusteral omega multiple sequence alignment. Dear students, the 3D structure of HPR-KP from Enterococcus faecalis is not available. In module 17 on protein structure modeling, we used automated mode of homology modeling for 3D structure prediction based on a single template structure. However, in module 18 on protein ligand docking, we found that there are 3D structures of HPR from Mycoplasma, Staphylococcus aureus and Lactobacillus casei. Using more than one template for homology modeling results in better structure prediction. But to undertake homology modeling using more than one template structures, we need to use only closely related 3D structures. The multiple sequence alignment is used for construction of evolutionary tree or the evolutionary history for the extent of relatedness. The evolutionary history is presented graphically using a pictogram known as phylogenetic tree. The phylogenetic tree reflects the divergence of genes and proteins from a common ancestor and therefore may be used to select closely related multiple templates for interactive homology modeling of the protein structures. There are hundreds of programs available for phylogenetic analysis. For complete list of tree construction applications, kindly visit evolution site at Genomics Division of University of Washington. These programs follow different strategies for construction of a phylogenetic tree. Therefore, visit Wikipedia at the shown address for theory behind various phylogenetic tree construction methods. We'll use PhyML application, which is a freely available web-based tree construction program and is based on maximum likelihood methodology. FIML application is also a part of the robust tree construction application for the non-specialist, which is available at Phylogenetic.France, and constructs MSA using commonly used multiple sequence alignment application muscle. At FIML, we find that FIML application requires pre-constructed MSA in Phylip format. We constructed multiple sequence alignment using cluster omega at UniProt knowledge base and downloaded multiple sequence alignment in FASTA format for the construction of web logo or sequence logo. Cluster omega multiple sequence alignment at UniProt knowledge base allows downloading multiple sequence alignment in FASTA format only. Therefore, we now need to convert the format of the saved alignment from the FASTA to the Phylip. Alternatively, MSA can be reconstructed interactively using cluster omega at European Bioinformatics Institute website. Visit format conversion site at the shown address. Choose the saved file in the FASTA format. Set the input parameters such as format to FASTA. Input format is FASTA, and select Phylip standard sequential as the output. And choose output line width as wide as possible and click submit button. In the ensuing window, the MSA in Phylip format appears. Click download hyperlink to display multiple sequence alignment in Phylip format. Copy it and paste it in Notepad and save the file with your given file name, but with the .phylip as extension name and choosing the save as type all files. The Phylip format has two sections, the header and the sequences. The header section has single line which records two numbers and an alphabet. The first number records the number of sequences in the file, 213 in the present example. The second records the number of aligned columns in the multiple sequence alignment. This includes the number of inserted gaps indicated with hyphen characters. This is 366 in the present example. And third is an alphabet to convey that all the sequences are entered in a sequential manner, that is, one sequence after the other in each separate line after the header section. The sequence line starts with an identification annotation typed in the first 10 columns. Therefore, if the identification annotation is shorter than 10 characters, as 6 in the present example, then this will automatically, the Phylip format will automatically append the remaining columns with 4 blanks, that is 4 spaces appended in the present case. Then to this identification annotation of 10 columns, the complete aligned sequence length of 366 including gaps in MSA is appended. This length is 366 in the present case. Then save this file in Phylip format with .phylip as extension name using Notepad as already explained. Now visit FIML, upload the saved MSA in Phylip format, set the type of sequence as amino acids, and set the type of Phylip format as sequential. Leave other settings to default. Enter a name for analysis, your email, then confirm your email and execute. The results link will reach your email ID in the indicated time. The ensuing page will confirm uploading and allow you to check the status and the results. The results link will also reach your email ID in the indicated time. Click the hyperlink to check job status. If the job is still running, it will be indicated. When the job is completed, click hyperlink, visualize your inferred phylogenetic tree. The tree is presented in the browser window. The tree can be saved in any of the image formats for printing. A typical bifurcating tree is shown here. The horizontal lines in the tree are called branches and represents the sequences belonging to the phylogenetic group or the family. Two adjoining branches are joined with a common ancestor at a node, which is represented with a circle in this diagram. The node on the extreme left, sometimes at the bottom in some tree programs, is called the root node, which represents the ultimate common ancestor of all members in the tree. The identities on the extreme right in this tree are existing sequences which we have used to construct the multiple sequence alignment and subsequently construct tree. All the nodes from these existing sequences till the root node are inferred ancestor sequences which do not exist today and are extinct. They are extinct sequences. The branch lengths are shown, and these branch lengths represents the evolutionary divergence times. The phylogenetic tree with this branch length is called a phylogram, but as biochemists, we are interested only in relationships, either close or distant, and the same may be represented through a dendrogram. Therefore, click dendrogram to display tree as dendrogram. In this tree, a dendrogram is presented in which all present day sequences used for constructing the tree are aligned on the right without mentioning branch lengths. As biochemists, we are interested in grouping sequences into closely and distantly related groups. Therefore, in this representation, each rightmost or the leaf node will have one present day sequence or a group containing more than one sequences with inferred non-existing ancestor sequences except when the other lineage is also present day sequence. One can search a node in the tree. Simply type the identification annotation in the search node text box and press enter key. In the present case, enter O07674 for HPRKP from Enterococcus faecalis and press enter key. The tree can be represented in linear or radial manner. Change the tree to radial manner by clicking radial radio button so that all leaf nodes, that is the sequences used for construction of the tree, are displayed in the minimum space. The search node will be highlighted in blue color. Click on the node to open context menu and select path to root command. This will highlight the path to the earliest divergence node in the tree in red color. Similarly, search other node you wish to map to the path. Enter Q9S1H5 for HPRKP from Staphylococcus xylosus. Then click on the node to open the context menu, select path to root command, and then this will highlight the path to the earliest divergence node in the tree in red color. In this way, users can estimate the divergence of the same enzyme in two species during evolution. Similarly, search for Q9RE09 for HPRKP from Lactobacillus casei. Then click on the node to open context menu and select path to root command. This will connect the path to the earliest divergence node in the tree. In this way, users can estimate again the divergence of the same enzyme in different species during evolution. Similarly, search Q8EWF5 for HPRKP from Mycoplasma penetrans. Then click on the node to open context menu, then select path to root command to connect the path to the earliest divergent node in the tree. So, you can estimate again the divergence of this enzyme in the species during evolution. This path to root will highlight complete path from root to these 4 nodes in red color. This highlighting reveals that divergence of HPRKP in mycoplasma from root occurred much earlier at second node than Staphylococcus, Lactobacillus and Enterococcus HPR-KP diverged at the recent 7th divergence node. This shows Staphylococcus, Lactobacillus and Enterococcus HPR-KP had a common ancestor at 7th inferred node of their common ancestor. Therefore, divergent template available from mycoplasma may be excluded from interactive homology modeling, and templates from closely related Staphylococcus, Lactobacillus may be used for homology modeling of HPR-KP from Enterococcus. Visit homology modeling at Swiss Model or Swiss PDB for interactive homology modeling at the shown address using Swiss PDB Viewer and Swiss Model Server, and follow the step-by-step procedure. Slide 58 is a video. This concludes the. Dear students, during evolution of proteins, functionally significant amino acid residues are conserved more than the rest of the sequence. Therefore, we learn to compile a set of enzyme sequences from Swiss Prot using keyword search from various species known to be related to each other either closely or distantly. Then we learned the construction of multiple sequence alignment from this set of sequences. The constructed MSA was displayed graphically as web logo or sequence logo to extract maximum amount of information hidden in the multiple sequence alignment. The multiple sequence alignment in the light of graphical display, that is sequence logo, was used to analyze to detect functionally significant residues from the multiple sequence alignment of HPR-KP. The conserved motif in the P loop for the binding of ATP or pyrophosphate and reactive diaspartate as well as another reactive histidine 136 and the transition state stabilizer arginine 202 could be predicted correctly in the consensus sequence by the cluster omega. Therefore, an MSA is a very useful tool for detecting functional residues in case enzyme substrate complex structure in 3 dimensions is not available. In addition, we learned construction of phylogenetic tree to detect closely related sequences, which helped in selecting multiple templates to be used during interactive homology modeling. I thank you all for visiting EPG Pathshala.